In the early 20th century, the world's health situation was far from perfect and faced numerous challenges. Issues related to sanitation and public hygiene contributed to the spread of infectious diseases, and global pandemics like the Spanish flu posed significant health threats. Limited technological advancements in healthcare infrastructure, along with restricted access to healthcare services, were also major concerns. Moreover, bacterial infections were the leading cause of death, but the available treatments were limited and ineffective. Medical knowledge about microorganisms and infectious diseases was still limited at that time, and surgical operations carried high risks due to frequent post-operative infections. This situation created an urgent need for an effective remedy against bacterial infections. Finally, in 1928, Alexander Fleming addressed all these health concerns by discovering penicillin. Alexander Fleming was born on August 6, 1881, at Lockfield Farm, near Darvel, in Ayrshire, Scotland. He was the third child of Hugh Fleming, a farmer, and Grace Sterling Morden. During his childhood, Fleming grew up in a simple rural environment, where he learned about life in the great outdoors. Fleming's early education took place at the Taradale Primary School, and at the age of 13, he moved to the local Kilmarnock Academy. It was at this school that Fleming's aptitude for mathematics and sciences began to shine. Despite financial limitations faced by his family, Fleming received a scholarship to study at the Royal Polytechnic Institution in London in 1895. After completing his education in London, Fleming decided to pursue medicine and enrolled at St. Mary's Hospital Medical School in London in 1901. There, he studied pathology and bacteriology and also received training in surgery. His extensive education in microbiology and bacteriology provided a strong foundation for the scientific research and experimentation he would conduct in the future. After finishing his medical education, Fleming worked as a bacteriologist at St. Mary's Hospital. He was interested in the risk of bacterial infections that commonly occurred in patients, particularly those caused by Staphylococcus bacteria. On September 3, 1928, at the St. Mary's Hospital Inoculation Department in London, Alexander Fleming conducted a simple experiment that would eventually lead to a groundbreaking discovery in the medical world. At that time, Fleming was investigating Staphylococcus aureus bacteria, which were a common cause of infections in open wounds. During his research, Fleming prepared several petri dishes containing cultures of Staphylococcus bacteria. After completing the experiment, he accidentally left the petri dishes on the laboratory bench during a long weekend holiday. At that time, London experienced warm and humid weather, providing ideal conditions for fungal growth. While Fleming was absent, some Penicillium genus fungi were carried by the wind or airborne particles into the laboratory and spread onto the petri dishes containing the Staphylococcus bacteria culture. When Fleming returned to the laboratory after the holiday, he examined the petri dishes he had left earlier. He was surprised by what he found. In one of the petri dishes, there was an area clearly free of Staphylococcus bacterial growth. This area was surrounded by a circle that prevented bacterial growth. Fleming realized that the accidentally introduced Penicillium notatum fungi had secreted a substance or compound with antibacterial properties. Fleming carefully observed and further studied this phenomenon. He realized that this fungus produced a compound that could inhibit the growth and kill Staphylococcus bacteria. He named this compound penicillin. This discovery caught the attention of scientists and researchers in the fields of microbiology and medicine. However, initially, penicillin was not considered a major breakthrough. Fleming continued his research on penicillin in an effort to identify the active compound that was effective against bacteria. However, he faced several challenges in developing penicillin into a clinically usable drug. Production of penicillin from Penicillium notatum fungi was limited in quantity for clinical use. Due to this limited production, it was difficult to meet the demand for this drug on a larger scale for widespread medical use. The pharmaceutical industry initially showed little interest in developing penicillin, as it was considered challenging to produce on a large scale and deemed economically less profitable. This hindered the progress in the development and production of penicillin. Furthermore, to prove the effectiveness and safety of penicillin clinically, further trials and research on humans were required. Initially, clinical research on penicillin was limited and not conducted thoroughly enough. The lack of clinical data kept penicillin in experimental stages and prevented it from being recognized as an effective medical therapy. In 1939, Howard Florey and Ernst Chain played a crucial role in the development of penicillin, leading to a significant breakthrough in the use of this antibiotic in medicine. They were part of the team at the Pathology Unit, University of Oxford, determined to develop penicillin for widespread use in treating bacterial infections. Florey and Chain took Fleming's research on penicillin to the next level. They succeeded in isolating and purifying the penicillin compound from Penicillium notatum fungi more efficiently than before. This isolation process allowed them to better understand the chemical structure and properties of penicillin. After successfully isolating penicillin, Florey and Chain tested its effectiveness in treating bacterial infections. They conducted experiments on mice and attempted to use penicillin to treat bacterial infections in animal subjects. The results showed that penicillin was highly effective in killing various types of pathogenic bacteria. Florey and Chain worked hard to develop methods for mass production of penicillin for clinical use. They increased the production efficiency of penicillin by optimizing the fermentation process using nutrient fluids. The next step was to test penicillin on humans to prove its effectiveness and safety clinically. Initially, trials were conducted on patients with severe and difficult-to-treat infections. The results demonstrated that penicillin was highly effective in treating bacterial infections and saving patients' lives. After successfully passing clinical trials, Florey and Chain collaborated with the pharmaceutical industry, including American companies, to mass-produce penicillin. Their efforts made mass production of penicillin possible, and the drug soon became widely available for use in medicine. In 1945, Florey, Chain, and Fleming, who had previously acknowledged the importance of Florey and Chain's role in penicillin's development, were jointly awarded the Nobel Prize in Physiology or Medicine for their contributions to the development of penicillin. The discovery and development of penicillin by Howard Florey and Ernst Chain revolutionized the field of medicine. Penicillin became the first commercially produced antibiotic, ushering in a new era in the treatment of bacterial infections. Their efforts in bringing penicillin from the laboratory to mass production saved millions of lives and forever changed medical practices.